So I'll be talking about cross-modal information fusion between SACS and EM, okay? And I'll try to make the case as to why that's a reasonable thing to do given the resolution revolution that's taken place in EM. But more generally, I'm interested in combining information across different experimental modalities in structural biology. Specifically in this talk between EM and small angle X-ray scattering, SACS. So this audience doesn't need any tutorial on EM. We have projection images through various directions. In small angle X-ray scattering, you have particles or structures in solution. It's an X-ray method rather than electron beam method. The solution conditions could be quite different than the conditions prior to vitrification in EM. And the mathematics of SACS is described in as follows. So if chi sub b is the characteristic function of your body, your molecule, think of it as the electron density. And if you perform the convolution of that with itself, actually with a reflected version of itself, and then decompose the position x into spherical coordinates and average over spheres, you get this pair correlation function, this PB of R. It's a radial function. Now, actually, that's not exactly what the SACS experiment provides, but that's the real space version of what the SACS experiment provides. In practice, what the SACS experiment provides is the Fourier space version of this, the intensity as a function of P. So I'm going to use P as the Fourier space radial variable, and R is the real space radial variable. And this sign of PR over PR is actually the zeroth spherical Bessel function. So I saw some posters yesterday. People were interested in Bessel functions. So those show up over here as well. So the information content in SACS is extremely low compared to EM. But nevertheless, we can use it in some ways, even just by itself. So this is work from many years ago that I did with Robert Jernigan and our collaborators on conformational interpolation, where we have two end states, two crystal structures. And we've adapted this method to take an initial crystal structure and drive it to the shape of a second structure using only the SACS profile curve of that second structure. Okay? So in other words, without knowing the full second structure, and actually we don't care about the pathway that it's taking. What we care about is, given an initial structure, how can we morph it to become consistent with partial experimental information about a second conformation. Okay? So this paper showed that it's feasible to take the SACS profile, the pair distribution function, by itself for a second structure, if you know the conformation of a protein in a different structure, how to morph it using that SACS information. So even though the information content is low in SACS, it can still be used. Now, how can we fuse that information, or why would we even want to fuse that information with EM? Well, as we know, in EM, you have your particles suspended in a thin layer of ice. The thinner, the better, except that when you get too thin, you're going to restrain the orientations of your particles. And therefore, you're going to have regions of projection direction which may not yield information. So what we can do, well, I'll go back here. What we can do is use SACS to fill in when there are too many missing projection directions in EM. That's the crux of this talk. But on the way, I'll talk a little bit about some other work that we've done in my lab. In particular, we worked on some class averaging methods, in particular for negative staining, which is a different story than EM, slightly different, but has a lot of commonality. And the take that we had on this was that when doing class averaging, you're trading off noise for blurriness in a sense. by stacking images and having the noise disappear. Because the images cannot be stacked exactly, that results in a motional blurring of the image relative to the underlying thing that you want to attain. So we developed some methodologies modeling this motion blurring as a convolution over the group of rigid body motions of the plane, and developed what kernel could be used to describe that blurring process, and developed a deblurring process by a deconvolution involving these methods. And these methods are built on work in my books. I'm actually not from this field. I'm from the field of robotics, but dabble in other things as well. And the connecting methodology between the various fields that I work in is this underlying math. And now, of course, these words, you know, convolution, Fourier, Gaussian distribution, that's all standard, except these books do it on Lie groups, such as the group of rotations or rigid body motions. Okay, so what do EM and SACS have in common? Do they have enough in common that it makes sense to fuse them together? And in the process of fusing, one needs to make sure that the two data sets are consistent, because the conditions in the SACS solution are very different than the conditions in the EM solution before vitrification. So they may be apples and oranges. That's why we need cross-validation. We need to make sure we're looking at the same things, the same conformations, before we try to fuse them together. Otherwise, it won't make sense. So there is complementary information. It's all the data is in Fourier space in the classical sense, not the Lie group sense. And so we use the Fourier slice theorem as everybody does. So the projection in real space corresponds to a slice of the Fourier transform of the density as seen in Fourier space. So we can use that. Now what the SACS information is doing, so this is we're going to step back and look at a 2D toy model as opposed to a 3D model. So the densities here are 2D densities on the plane, the projections are projections onto lines, the slices are lines, etc. And instead of spherically averaging as we would in SACS, we're going to average around the circle just to get started. Then we'll get to the 3D case shortly. But I should say from here on, I'm not going to use any real biomolecular structures. I'm going to do toy models, and I welcome anyone who'd like to work together on using this on real experimental data, both EM and SACS, and I'm happy to see you have a synchrotron, and so you can do both. Okay, so here's what happens. So we have this 2D density in the real plane. We convert it to Fourier space, and then we divide that 2D Fourier density into concentric circles, and we just look at the values on those circles. When we take these slices, we're taking slices through the center of these circles, so the actual data that would correspond to EM is at the intersections of these slice lines and the circle, and the SACS data is the integral of the square of this, or the modulus squared of the density around the circle. That's this 2D analog of SACS and EM. So if you're missing a big wedge of projection directions, okay, you can complement it with the SACS, okay? So in principle, these can be fused. In practice, we have to first check for consistency. So how do we check for consistency between the EM and the SACS data to make sure that the conformations, or even the ensemble of conformations in the SACS solution and the ensemble of conformations in EM are consistent with each other before we try to fuse? Well, that was this paper that we published a couple of years ago. And so I'll just briefly talk about how we do that, and then I'll get to the actual fusion process. So basically, what we can do is take the EM images, the slices in Fourier space, and divide those slice images into circles. We can average over each of those circles, and then average over all of the projection directions. So we get a P of R function for this data where it's P of R for circles, not for spheres, okay? And then we have the P of R function on spheres, the paired distribution function from Sachs. And what we showed in this paper actually is that you can use something called the Abel transformation to relate this data, which is sort of like you're projecting from the sphere to the circle, and do the inverse Abel transform. So basically we can compare the P of R functions that is generated computationally from the ensemble of EM data with the Sachs data, just to make sure at least that those P of R's match each other. Okay, but of course, one is on the circle, one is on the sphere. But using this Abel transformation, we can relate those two together, and if they match, that's the consistency check. That's the best that we can do. So the paper describes all of the details of why this is the right thing to do to relate this circular P of R with this spherical P of R. Okay, so now let's suppose that our data are consistent between EM and Sachs. How would we go about fusing them? Okay, so we're in polar coordinates in Fourier space. If the function in real space is real valued, then that places constraints on the value of the Fourier transform. And what we're doing in Fourier space to this, the planar, well, actually, I'm back to the 2D case. So this is data on lines going through the origin in Fourier space corresponding to our slice theorem. When we look at circles centered at the origin, we're going to develop a Fourier series in Fourier space of the Fourier transform, if that's not too confusing. And those Fourier series coefficients have to have certain symmetries to them that also originate from the real valuedness of the original density function. And then the intensity, okay, which is what we see in the Sachs experiment, again, this is in a planar analog, will just be the sum of the squared of the moduli of the Fourier series. Now, in EM, what we're getting is essentially the Fourier coefficient. We're getting the Fourier transform values on these circles where these projection lines intersect with the circle. And so basically we can expand a Fourier series around each circle of each radius in Fourier space. And I'm just here, we're going to just say for radius 1 just to make life easy, but it's the same for all, the same procedure for all radii. And then we're going to take this expression and we're going to manipulate it a little bit so that on one side of the expression is all the measured data, and on the other side of the expression are the variables that we want to obtain. So, for example, the Fourier values going around the circle in Fourier space, that's actually what EM is providing in this planar analog, okay? But it's slightly under-constrained, okay, because we have 2n plus 1 variables over here, the Fourier coefficients in Fourier space going around the circle, and we have 2n values over here. What we're going to do is we're going to take this center column, which is all ones, out, and we're going to treat this zeroth, the constant Fourier coefficient going around the circle, as a variable, okay? And so we're going to pull this over to this side, and then we're going to back substitute in and try to solve for that. But how do we solve for that? We're going to use the intensity from EM over here, from Sachs over here. We have this intensity value from Sachs. We substitute all of this in. The only unknown here is this rho 0 tilde, which is the constant value of the Fourier series going around each circle in Fourier space. And you know what? This is just a quadratic equation. So we solve for this unknown value, this rho 0, just solving, you know, the quadratic equation from junior high, okay? And then we put that back in, and here's a toy example. We're just looking at one circle where we know the function going around the circle, and we assume that we have theta values going around the circle in just at these specific angles. So these are the projection directions. Those are the only projection directions. And then we ask, how well can we reconstruct this, the Fourier series describing this function? The original function is blue. The reconstructed one, which would be analogous to EM by itself, is the black dotted one. And then we have two roots of this quadratic equation. If we choose wisely, we get the green, and then boom, it matches exactly the original blue. If we choose poorly, the root poorly, we get this wild red thing, which is worse than not fusing at all, okay? So we need to choose the root wisely. So we have a way to do that. And so now, this is as real as I'm going to get. This is my density, okay? I'm not looking at ribosomes. I'm not looking at, ion channels. We're looking at smiley faces. And this smiley face density is constructed as a sum of many, many Gaussian distributions, okay? And so, and of course, Gaussians in Fourier space have closed form solutions. The Fourier transform is closed form. And now what we're going to do is we're going to do a pretend, it's pretend at several levels because we're still density in the plane rather than 3D, but we'll get to 3D. And we're going to do some pretend EM projections by, in Fourier space, doing slices at different angles. And of course, if we sample going around enough, in this case, 65 angles going around, then we can reconstruct our smiley face very well, just as is done in computed tomography, in medical imaging. Now, but what if we have a wedge of missing projection directions? Okay, this is in Fourier space. We have this, and whenever I say there's a wedge, it's, of course, a two-sided wedge. If you're missing this, you're also missing the other side. And likewise in 3D, when we're going to have cones of missing projection direction, it's going to be a two-sided cone. So this is in Fourier space. If we use the slice theorem and we have these missing wedge, then our smiley face doesn't look so happy anymore, okay? Over here, this is EM by itself, and that's EM with Sachs, which I'm not bragging that that's so great, but you can see that there's some improvement, okay? Now, let's look at some other examples, okay? Here, we're missing these angular ranges as well as the other side of them, okay? So we're missing 30 degrees, so 60. We're missing 120, did I do that right? 60, yeah, 100. We're missing 120 degrees out of 360 here. If we try EM by itself, we don't get much, okay? If we do EM and Sachs, we get at least, you see the, there's eyes, there's the boundary of the face, okay? So, and over here, there's another example. So here, both of them basically don't work. I mean, you can still see some outline of the face. Over here, in just the projections, you don't get very much. Over here, this is the fusion. Over there, that's EM by itself. Now, remember that there's an intensity. The eyes were yellow. That was a higher intensity before. So even though the EM just visually doesn't look bad, the intensities are off relative to what they should have been given the initial model. Okay, now there are cases when they both fail, okay? And there are cases where adding the Sachs actually hurts. But most of the time that we've observed, adding the Sachs helps. Okay, so now let's go to move on to 3D. So when we do 3D, the slice theorem in 3D, we're getting planes of data through the origin in Fourier space. Those planes of data in Fourier space, we're going to divide Fourier space into concentric spheres because we need to do that for the spherical averaging in Sachs. So we're going to get intersections of spheres with planes through the origin. And so we're going to have data. Each EM image will give us a great circle of data on this sphere, okay? And then we have these concentric spheres. Okay, so then we need spherical harmonics. and other things, which, I won't give I think everybody here knows about spherical harmonics, and if we transform, if you have a spherical harmonic and you rotate it, you can re-express it in spherical harmonics, and the weights in that re-expression are the Wigner D matrices, or Wigner D functions, the entries in the Wigner D matrices. And so we use that, but first let's back up. If you have a real valued function in real space expressed on these concentric spheres, then these coefficients in the expansion in Fourier space are going to have this symmetry again. And the intensity is just going to be related to the modulus squared of these coefficients summed over the appropriate indices. And now we're going to do in 3D space what we did in the 2D case before, where I had that column of ones and I extracted out this row 0 and solved a quadratic equation. We're going to do all of that again, except now the entries in these matrices are spherical harmonic values at the unit vectors on the unit sphere where you have data, okay, corresponding to the slices in Fourier space. And, the math is not, it's not very sophisticated here. We again get a quadratic equation, and on the right side we have the intensity from Sachs. Everything else other than the row 0 on the left side of the equation is known from the EM the experiment, essentially from the slice theorem. In this case, the matrix F is not a square matrix, so we use a pseudo-inverse, and we get results like this. Now we have a 3D smiley face, okay? It has thickness in the z direction. If we look in Fourier space, we have concentric spheres again. We can, that's how we're going to visualize the data. Now, suppose that we have the green here corresponds to all the, let's say we have many, many projection directions. Okay, you have a rich set of projection directions, then there should be no surprise that we can reconstruct the smiley face. That's what, that's what you folks do day and night in reconstructing high quality densities from EM. But what if we have a very restricted set of projection directions? So here we've randomly sampled on the sphere, so those are our projection directions, and we're only keeping the ones in the blue cones. So we have a very limited number of projection directions. Then the data that we have on the sphere is going to be, each projection direction has a great circle of data associated with it. And that's what's shown on the right here. So we have relatively sparse data. What if we try to do a reconstruction with relatively sparse data? Well, if we use EM by itself, our smiley face looks like this. These are slices in different values of z, and this is looking down a projection, so you see the overall density. Again, it doesn't look too much like a smiley face. You get the boundary. Over here at this slice, you get the mouth. Over here, whereas there's a lot of junk in here, when we fuse with Sachs in 3D, there's less extraneous junk in here, and these slices look a bit better. Okay, so this is kind of like a proof of concept. If we're not worried about noise, if we're not worried about CTFs, if we're not worried about various things, then it looks like EM and Sachs, or EM augmented by Sachs, in the case when you have a lot of missing projection directions, can help. And that's the takeaway message from this talk. Here's another case where instead of drawing samples from the blue cone regions, we're drawing samples now from just the yellow regions and excluding the cone regions. And if you add up all the data from all of the great circles, it looks like that over there. And so in the simulated EM by itself, you get something that looks like this. Slices, some look like a smiley face, but others don't. And when you look at the projection down, you get this sort of messy stuff in the middle. when you combine EM and SACS, you get something that's somewhat cleaned up, okay? And then we did some other some other case studies. In this one, if the amount of data on your great circles is restricted to 1.5 times the radius, rounded off to the nearest number, you get kind of a mess over here, and it gets greatly cleaned up over there. And in part, that's because remember that SACS is physically integrating over the whole sphere. Okay, that's the theory of the experiment tells us that. So it's combining data. So if we have undersampling issues, then they can get cleaned up over there. And then here's another case. So, well, maybe the smiley face wasn't real enough for you, so then we did a Minion from Despicable Me, which may look a little more like a biomolecular structure. So here's, it has different density values. You can see the plot here. The body is low density. The eyes have black and blue. You get to higher density. And so if we do just a tomographic reconstruction, we do quite well, as expected, on the right. But if we restrict the projection directions, similarly to what I did for the 3D smiley face, then if we try to reconstruct using tomography or using Fourier slice theorem and back projection, you get something that's a bit blurry over here, but again cleaned up by SACS. The conclusion to take away here is that SACS and EM can be combined if we first cross-validate to make sure that we're adding the right conformations to each other. Here in these numerical experiments, we didn't have to do that because we had everything the same in both. It has potential, and that's all I'm trying to get across in this talk, is that there's potential to improve things when you have very restricted projection directions in EM and combining with SACS. And we're working to apply this on real macromolecular data. And the reason why I flew here halfway around the world from Singapore was to look for people who may be interested in trying this with real experimental data. I'd like to acknowledge the people who've worked on this, because there were a number of papers that I showed throughout this talk. Some of the folks worked on some of them and some on others. And this work was funded under an NIH grant. The last thing I'd like to say, well, thanks for your attention. And independent of whether you liked anything I talked about, you may enjoy some of the content in these books. And if anyone's interested, happy to send you a copy free of charge. I'll send it to you. So let's see how many takers I get. Okay, so if there are any questions, I'll be happy to answer them.